Yes. Good morning. Um, let's start the morning session. It's a great pleasure to introduce Dr. Glenn King. Glenn did his PhD at the University of Sydney before postdoctoral studies at the University of Oxford. After an academic period at the University of Sydney and the University of Connecticut Health Center, he joined the Institute of Molecular Bioscience at the University of Queensland in 2007. Glenn is a leader in the field of venoms-based drug and insecticide discovery. He early worked on spider venoms, led him to found Vestaron Corporation, an agriculture biotechnology company that developed the world's first peptide-based bioinsecticide. Glenn's current research focus on development of peptide drugs to treat cardiovascular and nervous system disorders. He recently co-founded Infenza Bioscience, a biotech company developing drugs for treating stroke and myocardial infarction. Glenn's laboratory maintains one of the largest venom collections in the world, sourced from more than 500 species of invertebrates. In 2023, Glenn received the Prime Minister's Prize for Innovation for his translational work, and in 2024, he was elected a Fellow of the Australian Academy of Science. Then, Glenn, thank you for being here to give your talk, and it's a great pleasure to listen to you.
Thank you very much for the kind introduction. Can you hear me?
Yes.
Can you hear me?
Yes.
Fantastic. Well, thank you for the kind introduction. Thank you for the opportunity to talk to you. I wish I could be there. As I think many people in the audience know, I love coming to Brazil. I was in Sao Paulo just a few months ago. And I'm also a big fan of fantastic architecture. And I love the work of Oscar Niemeyer. So I wish I was there with you in Brasilia. I'll just try and share my slides. Can you see my slides? Yes? Good. So for those of you that don't know what my lab does, we use arthropod venoms as a source of peptide drug leads for the treatment of human disorders in which ion channels play an important role in the etiology of the disease. And that probably raises two questions for you. One, why are we particularly interested in arthropod venoms as opposed, for example, to vertebrate venoms? And why are we particularly interested in ion channels? The reason we're interested in arthropod venoms to begin with is they're rich in small, very stable disulfide constrained peptides, which have many desirable properties of drug leads, as opposed, for example, to the small, less stable proteins that you typically find in snake venoms. They're also the richest known natural source of ion channel modulators. And so why are we interested in ion channels? Well, I think we're all aware that the most common class of human drugs are molecules that target G protein coupled receptors, but it's less well known that the second most common class of human drug targets is actually ion channels. A very large number of small molecule drugs target human ion channels, but many of them are very challenging to target with small molecule, hence our interest in targeting those ion channels with venom peptides. And we have three primary areas of interest. We're interested in visceral chronic pain, that is pain of the organs, and particularly the pain that's associated with irritable bowel syndrome and the pelvic pain that's associated with endometriosis. We work on a number of genetic epilepsies, particularly Dravet, KCNH1 and KCNQ2. But the area I want to talk to you about today is our work on acute cardiovascular disorders, and that's in particular trying to use venom peptides as leads to develop drugs for stroke, myocardial infarction, and heart transplantation. So I think this audience knows that cardiovascular disease is the #1 cause of death globally, and the most acute manifestations of cardiovascular disease are stroke and heart attack. And those two diseases alone, stroke and heart attack, are responsible for 27% of all deaths around the world every year. So each year about 9 million people die of a heart attack and 6.2 million people die of a stroke. And they're large numbers that I find difficult to comprehend. And so I think these numbers really drive home the importance of the problem. Over the next 24 hours, 24,000 men and women are going to die from a heart attack. Many of them will survive, but a large number of those will go on to develop heart failure. Over the same period of time, 17,000 women and men will die from a stroke. A similar number will survive, but we're left with a lifelong disability. And the reason there's such high mortality and morbidity associated with stroke and heart attack is we simply have no drugs at all to prevent the injury to the heart or brain that occurs during a heart attack or a stroke, none whatsoever. And that's the problem we're trying to solve. And I got particularly interested in an ion channel as a potential target for these cardiovascular disorders from this amazing paper that got published in Cell in 2004. What they did was they created a genetic knockout of a particular ion channel called ASIC1A, which I'll talk about in a moment, in mice. And then they gave those mice an ischemic stroke. And so the regular mice develop a very large infarct, which you can see here. And the heterozygous knockout mice, the infarct size was reduced by a remarkable 60% by knocking out that ion channel, yet the mice were otherwise phenotypically normal. So this looked like a really interesting target for, at least for stroke. So what are these ASICs? So ASIC stands for acid sensing ion channel. They're proton-gated sodium channels. There are 4 genes, 2 of which get alternatively spliced to create 6 subtypes of these channels: ASIC1A and B, ASIC2A and B, and ASIC3A and 4. They're expressed primarily, but I'm going to show you not exclusively, in the peripheral and central nervous system. And the functional channels are either homotrimers or heterotrimers. So this is the 3-dimensional structure of chicken ASIC1A, the one we're interested in. The 3 monomers are coloured differently. And you can see most of the protein lies extracellularly outside of the membrane. And the remarkable thing is this large amount of protein has evolved to recognise hydrogen ions. So the ligand for these channels is hydrogen ions. They respond to acidosis and they recognise protons. If you do a space-filling representation of the ion channel and map the electrostatic potential on it, you'll see in red here this deep acidic pocket. And that's the region of the channel that recognises the hydrogen ions and activates the channel. Now the subtype we're interested in is called ASIC1A. It's most prevalent in brain. In fact, it's virtually the only subtype present in human brain. And it's highly pH sensitive. So it's half activated at a pH of 6.6. And it's unusual for an ASIC channel in that it mediates the influx of calcium in addition to sodium, and that becomes important. Now, you may think this is a very extreme pH 6.6, but during a stroke and a myocardial infarction or a heart attack, the tissue pH can decline to less than 6.5. So during these 2 conditions, you might expect this channel to be persistently activated. So the question is, why does the pH drop so much in the heart and the brain after a heart attack and a stroke? So if we consider just a stroke for a moment, a stroke, an ischemic stroke at least, is caused by a blockage in an artery, which means that the affected region of the brain doesn't get blood or any oxygen supplied to it. Now neurons, just let me change my cursor here, it might be easier to see. Neurons are particularly energy-hungry cells. They require large amounts of fuel, and their primary fuel is glucose. So about 60% of the glucose that our body burns is burned by the brain, by the neurons. They can't burn the glucose oxidatively anymore, so they have to revert to anaerobic glycolysis. And for the biochemist in the audience, you'll know that the end product of anaerobic glycolysis is lactate. It's pretty much the same thing that happens when you're working very hard in the gym or exercising very hard. You can't supply oxygen to your muscle fast enough. You start to do some anaerobic glycolysis and you build up lactate in your muscles, and that's what causes that pain when you're exercised. But of course, your muscles are perfused, you get rid of the lactate quickly and the pain goes away quickly. But when you have a stroke, the artery's blocked and you can't remove that lactate. So the lactate starts to build up and the tissue pH starts to decline well below pH 7. And so after the stroke, when you get this tissue acidosis, that activates this ASIC1A channel. And that does several things. Firstly, as I told you, it brings calcium into the cell, and that will start to increase the calcium overload in the neurons. But also the sodium that comes in will depolarize the neurons, activate voltage-gated calcium channels, which brings in more calcium. And this calcium leads to apoptosis. But the channel also recruits an enzyme called RIP1 kinase, a master regulator of necroptosis, and sets off the necroptosis pathway. So the activation of the channel sets off two cell suicide pathways, both necroptosis and apoptosis. And the neurons begin to die, and that causes injury to the brain. Now, you may think when you go to hospital and the clinicians remove the blockage in the artery that the pH would restore pretty quickly. But it turns out that deuterium MRI has now shown that even after the brain is reperfused, it takes more than 48 hours for the lactate levels to come down. In other words, the ASIC1A channel is going to be persistently activated for a very long period of time after onset of the stroke. And so our simple hypothesis was, if we could find a good pharmacological inhibitor of ASIC1A, would that stop these suicide signals being sent to brain neurons during a stroke? And of course, we turn to venom peptides, arthropod venom peptides, which of course are the best known ion channel modulators we have. And at the time, there was one really good modulator of ASIC1A that was discovered in Michel Lizdunsky's lab in France, known as PCTX1. It was from this beautiful tarantula called Summerpears cambridgei. And it inhibited the human channel with an IC50 of about 3 nanomolar. So it seemed like a really good candidate to test our hypothesis. And so we gave mice a stroke, an ischemic stroke, and then we delivered a single dose of this peptide, this venom peptide, and the dose was very small, 2 nanograms per kilogram, and we administered it intracerebroventricularly, so this was proof of concept. We were delivering it directly into the brain, and we delivered it 2 hours after stroke. And I'll come back to why that 2 hours is important in a second. But you can see in the vehicle-treated animals, they get a large stroke, as indicated by these dark regions in the coronal sections. You can see the cortical infarct is very large here. And in the animals that were treated with PCTX1, you could see we got a really fantastic resolution of the infarct. So you can see here the size of the brain damage in the animals that received the venom peptide was much, much smaller. We actually had already, at this stage, done a lot of work on the pharmacology of this venom peptide. We knew what the active residues were, and we were able to make a mutant that was just 2 residues, just 2 mutations, had the same 3-dimensional fold, but it had much lower activity on the channel. And you can see this disarmed PCTX1, as we call it, did not work. You can see it did not reduce the infarct size, and that told us that this peptide was working by engaging with this ASIC1A channel. That was the basis of its mechanism of action, which was really nice. And that's where we got really lucky. A serendipitous event happened in the laboratory. We had been working on this spider for a long period of time. This is the Gari funnel-web spider. It's found in this island north of Brisbane called Gari. It's the biggest sand island in the world. And in the northern end of the island, around a place called Orchid Beach, there's a big population of these Australian funnel web spiders. And this is a picture of us, me and a postdoc, trying to dig them up. They live in the ground. They make burrows that are lined with web, hence the name funnel web. And you have to dig down about 20 or 30 centimetres to find the spiders. And the local indigenous people have a wonderful name for this spider. The Butchella people call this Majja Nilinguran, and that translates as long-toothed spider, because they have these massive chelicerae. It's like long teeth. So I had a fantastic PhD student in the lab called Sandy Pineda from Guatemala. And Sandy was simply looking at why the evolution of what we knew at the time was one of the most complex venoms in the world. So the venom of this spider contains more than 3,000 peptides. And so she was doing proteotranscriptomic profiling of the venom. And she showed that the venom contains 33 different superfamilies of peptides and proteins. The proteins were very low abundance, the peptides were much higher abundance, and most of these peptides had what's called an inhibitor cysteine knot or knot and fold, the red ones, and I'll talk about that structure in a moment. And in this family here, this family 1, which is not very highly expressed, she found a really interesting peptide. And it's this peptide here called HI1A. And the reason Sandy thought it was interesting when she came and showed me was because it looked a lot like PCTX1, which we had just shown worked really well in the context of an ischemic stroke. But it was twice as big as PCTX1. So what I've done here is shown one molecule of PCTX1 and then a second molecule of PCTX1 at the top here. And I've aligned that with one molecule of HI1A. And so you can see HI1A evolved by a tandem duplication of a PCTX1-like molecule. And what was interesting is we knew, as I told you before, what the active residues of PCTX1 were, so were the pharmacophore residues, and they were conserved in the first half of HI1A. So we suspected it was going to have activity on ASIC1A, but the question is, what did the second half do, which was less similar to PCTX1? Now, we had a bit of a problem, though. This was a huge peptide. It's 75 residues, and it has 6 disulfide bonds. And so if you do the mass, it turns out you can pair up these cysteines, the 12 cysteines, in 10,395 different ways. So the question was, how were we going to make the peptide with the correct 3-dimensional fold? Unfortunately, I had another talented PhD student in the lab at the time, Irene Chazignon from France, and Irene decided to try and make this by expression in the periplasm of E. coli, and she was very successful. She was able to make isotopically labeled protein or peptide, and we're able to determine the 3-dimensional structure. And this is the NMR structure over here. And you can see, as we expected, it contains 2 very similar domains, PCTX1-like domains, and each of these domains has a particular structure known as an inhibitor cysteine knot or a knot and fold, each of which has 3 disulfide bonds. And for those that aren't familiar with these knot and domains, they have very, very peculiar properties. So let me explain what they are. So each knot and domain contains a beta sheet with 2 or 3 beta strands and 3 disulfide bonds. And if we look at the top disulfide bond here and the bottom disulfide bond and the intervening sections of the polypeptide backbone, you can see they form a loop. And the third disulfide actually dives right through the center of the loop, and you can see that better in the video. So the orange disulfide dives through the center of the loop and effectively ties the polypeptide chain in a knot. And what that does is give these peptides, which are very common in spider venoms, incredible stability. And so if we take this peptide and we set it at 37 degrees in human serum for not 120 minutes, but 120 hours, so this is 5 days, you can see there's hardly any degradation. So this is stable as a rock in human serum, and that's not peculiar to this peptide, it's peculiar to this class of peptides. So these knotted peptides are usually very resistant to both high temperatures, solvents, and in particular to proteases, which is obviously fantastic from a therapeutic point of view. So how do we test whether this peptide has any activity on this ASIC1A channel? The way we do that is we harvest eggs or oocytes from the African clawed frog, Xenopus laevis, and then we inject messenger RNA encoding for the ion channel into the eggs, and we wait a couple of days for them to be expressed on the surface of the eggs. And then we can clamp them, do what's called 2-electrode voltage clamp. And then the way that we activate the channel, remember it's an acid-activated channel, so we have the oocytes sitting at pH 7.5, and then we drop the pH to 6, and you can see we get, in this case, a massive sodium current, so that's this inward deflection as a sodium current. And if we add just 100 picomolar of this peptide, you can see we already get a very big inhibition. So this is clearly a very potent inhibitor of the channel. If we do a full concentration response curve, we find it inhibits both the rat and the human channel with an IC50 of about 500 picomolar. I haven't shown the data here, but it's also incredibly selective for ASIC1A over the other ASIC subtypes, so it doesn't touch ASIC1B, for example. And more recent data suggests that the IC50 is actually closer to 200 picomolar. And to give you some context, there's one small molecule, the diuretic drug amelioride, that's very widely used as an inhibitor of this channel. And this peptide is 50,000-fold more potent than amelioride. So this venom peptide, straight out of the venom with no modification, is an exceptionally potent and selective inhibitor of this channel. The other thing that's really nice about it from a therapeutic point of view, if you, say, inhibit the channel on the oocytes with PCTX1, the peptide I told you about before, and then you perfuse the oocytes to try and wash it off, you can see you can recover the currents in about 20 minutes. This green line here. But if we try and do the same thing with HO1A, you can see it's very hard to get the currents back. In other words, it's very hard to wash the peptide off the channel. And in fact, we now know the off rate of this peptide from ASIC1A is many hours. And that's consistent with the fact that we couldn't really measure the affinity using SPR because it was less than 50 picomolar. So this binds to the channel with exceptionally high affinity, and it's very hard to wash off, which again, therapeutically is very good. So how does it work? Well, this is some great experiments done by Angelo Keramitis in Joe Lynch's lab. This is single channel electrophysiology, where you're able to take a small patch of membrane. So what we do here is we transfect hex cells with the ASIC1A channel, take a small patch of membrane that's got only one channel. So you can see the currents here are really tiny. The scale here is 2 picoamps. So you can see, and the downward deflections here indicate that the channel's turned on. So if we don't have the peptide present and we drop the pH from 7.4 to 6, you can see the channel turns on instantly, pretty much straight away, and it stays on. But in the presence of the peptide, the channel does not want to turn on. So what this peptide is doing is preventing activation of the channel, and that's exactly the mechanism we were looking for. Because remember, I told you before, when the channel activates, it immediately recruits RIP1 kinase and causes the neurons to commit suicide. So that was the desired mechanism of action. So that was nice therapeutically as well. So the question is, does it work in stroke? Does it actually do anything? And so we tested in this really nice model of stroke that you can induce in conscious animals. So we take spontaneously hypertensive rats and cannulate them a week before the experiment, and then a week later, you can infuse endothelin-1, a vasoconstrictor, into the middle cerebral artery. And that occludes the artery and causes a focal stroke. And the nice part about this model, it avoids the confounding factors from surgery and anaesthesia that you get in the more common model where you actually insert a filament into the artery. So we think it's a nice model. And I'm not going to go through the data because it was published some time ago, but simply to say that when we deliver, again, a very small dose, similar to what we did in the PCTX1 experiments, ICV, 2, 4, or 8 hours after a stroke, we get remarkable reduction in infarct size. Now, the reason these time points are really important is many drugs, or all drugs that have been trialled in clinical trials for stroke have failed. One of the reasons many of them have failed is the drug was initially delivered either before the stroke, just after the stroke, or within the first hour after the stroke. And that's not really clinically relevant. If you get to a hospital within the first hour or so after your stroke, there's a good chance that they'll be able to clear that and you'll be fine. 60% or more of stroke victims take over 2 hours to get to the hospital, and they're the ones that are in serious trouble and the ones that you need to treat. So your drug needs to work at 2 hours and longer after stroke onset, otherwise it's really not clinically very helpful. And so we showed that at 2 hours and 4 hours, it reduced the infarct size by 80%. And even when we went out to 8 hours after stroke onset, it still reduced the infarct size by 2/3. And there were concomitant improvements in the neurological performance of the animal and their motor performance, as you might expect from this really incredible decrease in infarct size. So why do we think this is really important in the context of treating an ischemic stroke? Well, there is one drug that you can potentially use to treat somebody who has an ischemic stroke. It's called tissue plasminogen activator. It's an attempt to enzymatically remove the clot. The problem is there are two types of stroke. There's an ischemic stroke, which is due to a blockage in an artery, and there's a hemorrhagic stroke, which is a rupture of an aneurysm causing bleeding in the brain. And the problem is you can't use TPA for a hemorrhagic stroke because it could potentially make the situation much worse. And what that means is the first responder, who doesn't know whether the person's having an ischemic stroke or a hemorrhagic stroke, cannot give the drug. And what that means is that when patients are being transported to hospital, they're losing about 2 million neurons per minute after the onset of the stroke, and there's nothing you can do about it. The advantage of this venom peptide with this mechanism action is you could potentially deliver it by first responders because it would be fine for people with a hemorrhagic stroke. It shouldn't cause any problems. But even if you can't deliver it early, we've shown that even if you take 8 hours to get to the hospital, you can still get benefit. And of course, it's compatible with current reperfusion strategies. It's because the peptides had such a profound effect in the context of an ischemic stroke, we wondered whether the channel might be important for ischemic injuries of other organs. And so I think this graph is really instructive because what it shows is how long you can keep an organ for after you take it out of a donor before you have to transplant it. Excuse me. And you can see you can keep a kidney, for example, for several days. But after the brain, the organ that's most susceptible to ischemic injury is the heart. You can only keep a human heart for up to 4 hours. What that means, for example, if you had a donor up in Recife, you're probably not going to get it to a recipient in Sao Paulo in time to transplant it. There's just not enough time because the heart's not going to last long enough. So we wondered whether this channel might play a role in ischemic injury of the heart. And actually, when we looked at GWAS, when we looked at some GWAS data looking for correlations between polymorphisms in the ASIC1A gene and various diseases, we found a correlation with ischemic stroke, which we expected based on the data I just showed you. We also found there was a strong correlation with myocardial infarction, which made us think maybe this channel does have a role in ischemic injury of the heart. So the question was, is it even there? And so I got lucky in that soon after we published the stroke work, we had a new group leader arrive at our institute called Nathan Palpant. And it turned out Nathan is an expert in cardiovascular physiology. And so I sat down and explained what we'd found in stroke and asked him what was known about this channel in the heart. And he said, well, nobody's ever looked to see if it's even there. We don't know if that channel's present. And so we were able to do transcriptomic profiling of both mouse cardiomyocytes and human iPSC-derived human cardiomyocytes. And we showed that, in fact, the ASIC1A channel is present in human heart. The other subtypes are not present. And so that was really nice. That was the first time anybody had really shown the channel was present in human cardiac muscle. So the question is, does it have a role, though, in ischemic injury of the heart? And so we use this really nice model of ischemia reperfusion injury called the Langendorf ischemia reperfusion model. Some of you probably use this model. You take a heart, it could be a rat heart or even a pig heart, and you retrogradely perfuse it through the aorta to keep all the valves open. And you can keep that heart, as long as you perfuse it, you can keep that heart beating for hours. And so this is actually a rat heart on a Langendorf rig, beating at several hundred beats per minute. And you can keep the heart beating like this for several hours. Of course, you have complete control over the perfusion of that heart. So you can actually stop perfusing that heart and give it a global ischemia. And so that's what we did. So we did that, took mouse hearts in this case, put them on a Langendorf rig, and we used hearts from wild-type mice and mice in which we created a very specific ASIC1A knockout. And then we turned off the perfusion for 25 minutes and measured a whole bunch of parameters. This parameter here, LVDP, is left ventricular developed pressure. It's one of many parameters we measured. And as you might expect, after we turn the flow off, that goes to 0. And then we turn the flow back on and ask the question is, can we recover any function in that heart? And here's something really interesting happens. We just look at the wild-type mice first of all in the grey. You can see they start to recover very quickly. And then the LVDP suddenly drops dramatically. And that's the reperfusion injury. So the paradoxical thing is to recover the heart of somebody who's having a heart attack. You have to reperfuse the heart. You have to remove the blockage and reperfuse the heart. But the paradox is the act of reperfusion itself, rapid reperfusion, actually causes additional injury to the heart. So the standard of care actually induces additional injury. So that's the reperfusion injury, and then the heart slowly starts to recover. But the really nice thing was we found the recovery was better in the hearts where we knocked out the ASIC1A channel. And so then we tried a venom peptide, of course, same experiment, except we're using wild-type mice plus or minus the venom peptide HI1A, and you can see the hearts that got the venom peptide recovered much, much better than the hearts that didn't. So these experiments combined suggested that ASIC1A did play an important role in ischemic injury of the heart. And so then we went to a full in vivo model of myocardial infarction. It's a pretty standard model where you ligate the left anterior descending artery. So that's this artery here. It's often known as the widowmaker, and the reason for that is it supplies about half of the arterial blood to the left ventricle. So if you get a major blockage in a left anterior descending artery, or the LAD, you need to get to hospital very quickly because otherwise there's a big chance you could die, hence the name widowmaker. So we ligated the LAD of mice for 40 minutes, and we delivered the venom peptide HI1A 5 minutes before we started ligation, 5 minutes after we started ligation, or 5 minutes before we reperfused the heart, that is, took away the ligation. And then we followed this up with echocardiograms and histology out to 1 month, and we measured infarct size, of course. And what we found when we delivered the drug, and this was now delivering the drug intravenously, so IV, at 1 mg per kg in the mice, whether we delivered it 5 minutes before, 5 minutes after, or just before reperfusion, didn't seem to make much difference. In all cases, we got a very significant reduction in the infarct size. There was a nice dose dependence. When we went to 0.5 mg per kg, we didn't lose much efficacy, but we lost it almost completely when we went to 0.1 mg per kg. We looked at fibrosis at 1 month. The animals that got 1 mg per kg venom peptide had significantly reduced fibrosis. The reason that's important is you may not know that the human heart has virtually no capacity to regenerate. So an amazing statistic is that when you die, about 50% of the heart muscle cells you have are muscle cells that you were actually born with. So those muscle cells can have to last for 80, 90, 100 years. And so when you lose heart muscle cells during a heart attack, there's very little ability to recover them, and so it becomes fibrotic. So the level of fibrosis is an important measure. A really important measure of heart function is called the ejection fraction. It's how much blood the left ventricle pumps out with each contraction. And when that dips below 40%, there's a good chance you're going to progress to heart failure. So you can see the mice that didn't get the drug, you can see they've got a much reduced ejection fraction compared to the sham animals. But the mice that got the HI1A, their ejection fraction remains pretty good. So we've got less infarct size, lower infarct size, less fibrosis, and a better functioning heart. You may ask, why did you bother to deliver the drug 5 minutes before you did the ligation of the LAD? Because you don't know when you're going to have a heart attack, right? So what's the point of that experiment? Well, the point of that experiment is there are certain cardiac surgeries where you know you are likely to cause an ischemic injury to the heart, and you might want to deliver it before the surgery. And a classic example is coronary artery bypass graft surgery, where you're going to put the heart on bypass and you probably are going to give it an ischemic injury. So you could deliver the drug before the surgery. The 5 minutes after would be is mimicking first responder giving the drug after the patient is first found. And the 5 minutes prior to reperfusion is mimicking the situation in the hospital where you try and remove the blockage and reperfuse the heart. In that stage, the heart transplant group down at the University of Sydney, one of the best-known heart transplant units in the world, found out what we were doing and contacted us about checking whether our molecule might be helpful in terms of extending the life of donor hearts. And I told you before that's a big problem because they only last for 4 hours. And so I knew nothing about heart transplant at the time. And so I asked Peter McDonald, the head of the transplant team down in Sydney, how do you treat a human heart when you take it out of the donor? And he said, oh, we just stick it in an ice box. And I laughed and said, no, what do you really do? And he said, no, that's what we do. We stick the heart in a buffer and we stick it in an ice box. So that's the standard of care for a human heart that's about to be transplanted. And I was flabbergasted by that. And that's why it only lasts 4 hours. It has no oxygen supply, it has no blood, it's just sitting there on ice. And Peter, so the experiment we did was this. We took rat hearts, we took them out of a rats as though they were going to be donated, and we put them on one of these Langendorf rigs and we measured their function. So we had a baseline. And then we stored them. Now, I told you that these hearts usually only last for 4 hours. So we stored them for 8 hours with the drug. And we were using just 10 nanomolar of the venom peptide. And then we put them back on the rig after the storage and we measured their parameters again so we could compare it to the baseline. And we measured a bunch of parameters. I'm just going to show you a couple. So this is the aortic flow. And these are hearts in red that were just put in the preservation buffer called Celsia. And you can see they're pretty much dead. They're not they wouldn't be transplantable, right? There's no aortic flow. That's exactly what we expected. But you can see the hearts that got the drug, the HO1A, were pretty good. They had good recovery of aortic flow and good recovery of cardiac output. So this was a fantastic result in that we were able to keep these hearts viable for up to 8 hours. Now, Peter has pioneered a new way of doing heart transplantation. Most heart transplants around the world are done using donation from brain-dead donors. But there's potentially another way, and that is to get hearts by donation after what's called circulatory death. And so these are patients who, unfortunately, after discussion with the family, are taken off life support. And the definition of death in that case is the absence of contractions, heart contractions, so that the heart is stopped. And it's been thought that you could never recover those hearts because, as Wikipedia says, asystole is the most serious form of cardiac arrest and is usually irreversible. And it's worse than that because not only do you have a heart now that you've got to wait for it to stop. Most jurisdictions around the world, I'm not sure what the situation is in Brazil, but all jurisdictions mandate you then have to wait a period of time to make sure the heart doesn't start again. In Australia, it's 5 minutes. In Italy, it's 20 minutes. It's quite a long period of time. So the heart is sitting there, it's warm, it's ischemic, it's dying, and it's always been thought those hearts are not recoverable. And I think this shows you the why that situation's so bad. So this is simulating the cessation of life support in a pig by tracheal ligation. So you withdraw life support, the heart gradually stops. So here it's basically stopped, but then you've got to wait this standoff period between 5 and 20 minutes. And you can see the heart is dead at that point. It's stopped. It's not beating. It's still warm though. It's in the body. And it's always been thought that you could never recover that heart for transplantation. But what Peter showed is you get that heart out quickly and you rapidly perfuse it, give it blood, give it oxygen. As long as you do that quickly, you can resuscitate those hearts and transplant them. And in fact, in Sydney and in Australia and the UK now, about 30% of heart transplants are done using this method. So it's been a really profound change in the way heart transplant is done. But what Peter has shown, that period between what we call the warm ischemic time, between withdrawing the life support, getting it out and getting onto the perfusion rig, when that gets beyond 15 minutes, the heart becomes untransplantable. So this depends a lot on the skill of the surgeons to get it out, get it on the rig quickly. So what we did is we made this warm ischemic time 20 minutes, right? Which is longer than the period in which they usually can recover a heart for transplant. And this was done with pigs, I should say. These are pig hearts. Same type of experiment before, where you put it on a Langendorf rig and measure the performance of the heart. And you can see this is aortic flow over here again versus pressure. And you can see the hearts that didn't get the drug. So this is the sham. This is the hearts down here in grey. You can barely see them because they're all sitting at 0, are dead. So the hearts that got no drug, you could give them antiplatelets, you can give you anticoagulants, whatever you like, but they were dead hearts. But the hearts that got the venom peptide, again, just 10 nanomolar, were not that much different to sham, which was a remarkable recovery of these hearts, given that they were subjected to very extreme ischemia. So we knew at this stage we had a drug candidate that was potent, selective, and I haven't shown you the data, but very safe. We've shown activity in multiple disease models, efficacy in multiple species. We've seen efficacy in mice, rats, pigs, and sheep. We've shown efficacy at clinically relevant time points. We'd seen a strong genetic association between the ASIC1A gene and heart disease. Now, for those of you that don't know, when you can show a genetic association between the gene encoding your target and the disease you're trying to treat, your chances of successfully developing a drug for that disease increase at least 2 or 3-fold. And we think, importantly, the drug we're trying to develop targets what I think is the key pathophysiological event caused by cardiac ischemia or cerebral ischemia, which is tissue acidosis. And no other drugs really have tried to target that. So we decided to spin out a company to try and develop this molecule. And we raised the money entirely from private investors. And our focus is on initially a particular type of heart attack called STEMI. And the reason we focused on heart attack is simply drug delivery is much easier. So we can just go IV for all of the cardiac applications. The reason we're interested in STEMI, it's the most acute form of MI. It's very easy to diagnose in an ambulance. So this is a typical 12-lead ECG, and this is the ST segment here. And for somebody that's having a STEMI heart attack, ST segment is very elevated. And so you can pick it pretty clearly in the ambulance. The first responders know they've got to get that person to hospital very quickly because it's a very severe heart attack. And all these patients get pretty much the same treatment. So they get something called percutaneous coronary intervention, which is balloon angioplasty combined with stent implantation. So it's a well-defined patient cohort, and it's a fairly standardized treatment protocol, which is why we like that group. But of course, we had to show the drug worked for that extreme type of heart attack. And so we went back to that model I showed you before, but this time did a 90-minute ligation with delivery of the drug just prior to reperfusion, so mimicking the hospital situation. We also decided to benchmark the venom peptide against a drug called cariparide. Cariparide is a drug that made it all the way to phase 3 for coronary artery bypass grafting and myocardial infarction. And it turned out to be quite cardioprotective. But the problem was it caused significant side effects, including stroke, unfortunately. So it was withdrawn from development. But nonetheless, it's a good benchmark for us because it is cardioprotective. And you can see that in this really severe model of heart attack, our drug, which is the red one here, the venom peptide, is just as cardioprotective as cariparide, this drug that made it all the way to phase 3, which gave us some confidence that was likely to be translatable to humans. I want to finish the data by showing you the HO1A data by showing you this experiment. So we've now gone to a full transplant in pigs. using donation after circulatory death with an extreme warm ischemic time, so 30 minutes warm ischemic time. The human heart that was subjected to this would definitely not be transplantable. And so what we do is we take the heart, subject the pig heart to 30-minute ischemia, we take it out, we put it on the perfusion rig, and then we try and transplant it into a new pig, a donor pig, a recipient pig, and then try and wean that pig off bypass. And so the question is, over the first 3 hours, can we wean that pig off bypass? I'm just showing you the first experiments done with hearts that did not receive the drug. And you can see none of the pigs survived for more than 3 hours off bypass. So one passed away immediately, one lasted 1 hour, one lasted 2 hours. I'm just showing you the first 2 pigs here that got the drug, where the heart got the drug, the venom peptide, and you can see they both survived for the 3 hours off bypass. We've now done this with 6 pigs. I haven't got the data. All 6 pigs that didn't get the drug didn't survive, and 5 of the 6 pigs that got the heart with the drug survived. So this was a remarkable demonstration of the utility of this drug for increasing the viability of, in this case, pig hearts. And the reason we use pig hearts, by the way, is pigs had very similar hearts to humans, and so they're a very good model system. So we think HO1A potentially would be a good drug for applications in both myocardial infarction and heart transplantation, but the problem is for stroke, you have to get it into the brain, and HO1A is not going to have much brain permeability. The blood-brain, and that's because of the blood-brain barrier, which most of you will know about. There's a barrier around the cerebral blood supply that prevents the influx of most molecules into the brain. It's compromised after stroke, but that compromising doesn't begin until about 1 to 2 hours after stroke onset. So in other words, intravenous HO1A might be effective for patients that arrive in hospital, say more than 2 hours after their stroke, but it's probably not going to be suitable for first responders. And we'd really like to develop a drug that first responders could use, because as I said to you, you lose 2 million neurons per minute after the stroke starts. So the earlier you can get a drug in there, a neuroprotective drug in there, the better for the patient. Now, there is a drug, peptide, that made it into phase 3 recently for stroke, and they conjugated it to TAT, a cell permeable peptide, which many of you will know about, for getting it into the brain. We wondered whether there might be a better way to develop a blood-brain barrier permeant peptide for use by first responders. How am I going for time, by the way? So we teamed up with, we have been making the channel for structural biology studies. So we do have a cryo-EM and an X-ray structure of ASIC1A, which we've never published. But of course, we had the channel, we were able to team up with Hiroaki Suga at the University of Tokyo and apply this incredible approach called RAPID for trying to develop cyclic peptides that would bind with high affinity to the channel. The way this works is you have a DNA library of random peptide sequences and a very large number, more than 10 to the 12 peptides. You translate this to messenger RNA, conjugate it to puromycin, and then you do translation. So now you've got your peptide, your translated peptide, bound by puromycin to the messenger RNA. You do reverse transcription. So now you have a huge library of these little peptides, which actually cyclize after translation, attached to their encoding DNA. So then you just pull down against your target, in our case is ASIC1A, to find out the best binders, which we did, and it worked incredibly well. The advantage of this approach, by the way, is you can, because it's done in vitro, you can do genetic code reprogramming and you can incorporate non-natural amino acids, which we did in some cases. So I can't show you the sequence, but basically these are 14 residue cyclic peptides, so they're cyclized via this thioether bond, and that's a stable linkage, by the way. They bind ASIC1A, human ASIC1A, with very high affinity, so less than 250 picomolar KD. But here's the exciting part. What you're looking for here is binders. There's no guarantee that they will do anything. And there's no guarantee that they will do anything like the venom peptide. But when we looked at the sequences of this, these peptides, as I told you before, we knew what the pharmacophore of PCTX1 and HO1A was, and what we realized straight away is that we had in vitro evolved a small mimic of the venom peptide, a cyclic small version of the active site of the venom peptide. The advantage now is, though, that this is very easy to make. We've made more than 300 analogues of these small peptides now. They're very active on ASIC1A, they're very selective, they have a good safety profile, and what's nice is when you put them into that model I showed you before, and myocardial infarction, so this is with the 90-minute ligation of the LID, they're pretty much, for this peptide IB100, is equal potent with the venom peptide. And so, in fact, the company has now nominated its candidate for phase 1, which is one of these peptides, the wholly optimized one, and we should be able to start phase 1 studies in Q3 or Q4 next year. So to finish up, the take-home messages are that I hope I've convinced you that arthropod venoms can be a very rich source of peptidic ion channel modulators. In the case of spiders, most of these modulators are not in peptides with exceptional protease resistance, which is good therapeutically. Many of them, not all of them, but many of them have exquisite potency and selectivity for their ion channel target. We think HO1A is potentially a promising candidate for treatment of heart attack and heart transplant, but the rapid derived cyclic peptides, or mini HO1As, are likely better suited for the treatment of stroke. So I just want to thank the people that did the work, particular thanks to Sandy, who originally discovered the sequence of HO1A, to Ren, who was the first person to make HO1A and showed that it worked in the context of stroke, to Natalie in my lab, who has worked incredibly hard to make HO1A for pretty much all of the experiments I've shown you, to Nathan Palpat and his team for all the work in the myocardial infarction, for Peter and Sarah for all of the heart transplant experiments, Lachlan, Joe and Angelo for the pharmacology, and Toby and Hiroaka for the rapid screen. For those of you that want to know more about this, there was a science show on Australian TV called Catalyst that did an episode about this work, and you can go and look at it there. For those of you who want to know more generally about what we do, we have a very talented musician in the lab called Andy Walker, who wrote a song called Peptide, which is about venom peptides, and you can go and look at that at YouTube. I've been incredibly lucky to have many Brazilians in my lab, and this was a wonderful situation a couple of years ago where I had 4 in the lab at the same time, 2 of which were very talented musicians, and so this is one of our parties with Francielli on piano and Cyril on guitar, and Anna and Joe is here contributing to the singing. So I'll end there, and I'd be very happy to answer any questions.
Thank you, Glenn, for the amazing talk. And then we are
Welcome.
open. We are open for questions.
Hi, Glenn. Thank you very much for the amazing presentation. You showed that the ASIC receptor is present in many tissues, and I wonder if you tested leukocytes for that, because some of the responses of the ischemia reperfusion can be systemic, and this would be interesting.
I think that's a really good question. It is present. It's not present in many organs, so it's only present in kidney, heart, probably the eye, and the brain. But it is present in a number of immune cells, as you point out. And I'm not entirely convinced that the effect that we see in the context of stroke in particular is entirely due to the effect on the neurons. We may, as you suggest, be affecting neuroinflammation post-stroke as well. And that's something that I'd very much like to test. So it's a very good question. And yes, they could. The immune population of ASIC1A could be involved.
Thank you.
Hi, King. Maria Helena. Wonderful work. Last time I heard you had only the toxin. Now you have a nice peptide. And
Yes.
very nice. My question is, as these channels are widely distributed, have you observed some side effects? Or is a
good
perfect peptide?
Yeah, good question, Maria Elena. Not with the venom peptide. The venom peptide is really clean pharmacology, so we can dose that in animals pretty high. We can run all of the in vitro safety tests. You probably know about the safety screen 44 that many companies run, where you test it against 44 common, receptors, ion channels, and enzymes. It's very, very clean. The smaller peptides, as you might expect, are not as clean. So we've had to work a bit harder to get rid of some of the off-target effects of the smaller peptides. But we have done that, but there was one particular problem that we had to analog around. But now they're very clean now, so we've been able to dose. I think we have at least a 50 to 100-fold therapeutic window now. So I think we're pretty good.
Hi, Glenn, Gisele. Amazing talk.
Gisele, how are you?
I'm fine, thank you. And you? Amazing talk,
Thanks.
impressive job. My question is, you said that drugs that block ASICs have neuroprotective potential. Have you tried your peptides in neurodegenerative diseases?
We haven't, but Lachlan Rash, who you may know, Gisele, was a postdoc in my lab for a while, is the person who really introduced the ASIC project to my lab. And as Lachlan's been looking in the context of multiple sclerosis and seeing really good efficacy, and surprisingly seeing good efficacy with the venom peptide delivered IV, which is really surprising. But he's seen pretty good efficacy in the EAE model of multiple sclerosis, Gisele. I don't think anybody's looked at it in the context of, Alzheimer's or Parkinson's or any of those sort of neurodegenerative diseases, though. Because the one neurological area that I think is interesting is the only phenotype that the knockout seems to have is in relation to fear. So if you look at fear conditioning, the initial papers reported that the knockout animals were different. And interestingly, all of the students that I've had that have worked with the knockout mice, when I asked them, do these mice have any phenotype? Can you see anything? They all say the only thing is the mice are really happy. The knockout mice are really happy. You can do anything to them, which fits in with this idea that it's involved in fear conditioning. And so that's probably something worth following up.
Okay, thank you.
More questions? I have a question, but actually my name is Gloria. Sorry, I didn't introduce myself.
No worries.
Nice to meet you. You said in the beginning of your presentation that there are 4 genes for this channel, and some of them have alternative splicing isoforms. Did you check the binding properties of the peptide HI1A in different isoforms and alternative splicing isoforms of the channels? Do you think they bind as the same?
It's a good question. So there's only a couple of isoforms for which we know their role. So ASIC1A, B, 2A, and 3 are ones for which we know the function. So they're the ones we've tested our peptide against. And HI1A is really selective. It's got at least 1,000-fold selectivity for ASIC1A over those other subtypes, so 1B and 3. It wouldn't probably be clinically a problem if it inhibited 1B and 3, because they're involved in pain, so it would probably be analgesic if it inhibited those. But nonetheless, I think it's still good to have very clean pharmacology.
Mm-hmm. Great. And you also said that there are polymorphisms in the population that are checked by GWAS. Do you think the polymorphisms in the population, especially in Brazil, that are very different population and admixture population, do you think that the peptide will act as the same in not admixture populations?
Yeah, that's a great question, Flora, and it's something that we're really interested in, because the thing is, those mutations, those polymorphisms may make those people more susceptible. but that mutation also may mean your drug doesn't work anymore, right? Because it might be the binding site for your drug. And so what we've done actually is we've gone and made those mutant channels to see whether the drug still binds to those mutant channels. So we haven't found any yet where it's a problem, but we'll continue to test those, because I think the point you raise is a really good one, is you need to make sure, because they're going to be the people that need it the most, right? So it would be terrible if your drug didn't work in the people that needed it the most.
Yeah.
So we are making mutant channel. Yeah, that's a great question.
Thank you. I think we don't have more questions. And then thank you again for the great talk. Amazing. It was a nice pleasure to meet you, even online. Sorry. Bye. Bye-bye.
Thank you. Bye-bye. Bye.